artificial reproduction of African catfish, or Clarias gariepinus. The beautiful sunrise over the eastern horizon throwing a golden rays upon the length of Lake Tana. It creates a twinkle jewel on the water's surface. Thereby, a rooster with its croaking, kukololo, sound accompanied by morning bird's melody, heralded the beginning of a new day. Similarly, research with a novel idea, flawless objective and testable hypothesis, as it was supported by budget, getting a green light approval after a peer review, both phenomenal progress in their own direction. The Bahir Dar Fishery and Other Aquatic Life Research Center, under the Amhara Regional Agriculture Research Institute. Its main direction of research activity focused on artificial propagation of fish. In order to stock a newly built man-made water holding structure, natural water bodies initially devoid of fish and enhancement of those water bodies, which exhausted due to unwise utilization and small pond built by farmers for multipurpose utilization. Additionally, rehabilitation and conservation of the endangered endemic fish species of Lake Tana labiobarbacis fish species, also implemented through artificial breeding scheme, by rearing in large number and releasing to the lake. On this documentary film, the rearing of African catfish, or Clarias gariepinus, are tuned up according to the research plan of the center. By local name, Ambeza or K-Asa, or in its scientific name, Clarias gariepinus, is a widespread freshwater fish species found in Ethiopia, Middle East, and throughout Central and Southern Africa. It inhabits natural lakes, fish ponds, streams, and natural ponds in both deep and shallow waters. Generally, its natural behavior is suitable for aquaculture, thus, it is the most preferable culture fish species, next to Nile tilapia and common carp. Since the catfish flesh is fully clean of spiny rays, it is the most preferable for preparation of different dishes. This situation makes the catfish popular in different parts of the world, in Europe, USA, and Asian countries. So, they culture in large fish farm and produced in thousands of tons. Clarias gariepinus, in nature, will produce during the rainy season, mainly from July to August, following the inundation of the plain after flooding of tributary rivers. Following this flooding pattern during nighttime, the catfish migrate to the surrounding flooded plain and spawn. The reason is that the fertilized eggs have a sticky nature, so it needs grasses and other plants to adhere on it. This makes it difficult to find a catfish fry in nature. So, in order to get a catfish fingerlings, it has to be implemented in artificial fertilization and rearing of catfish. This documentary film tries to show the overall techniques of artificial rearing of catfish from the collection of pituitary gland until the production of fingerlings. Collection of pituitary gland. The pituitary gland extracted from fully mature fish of both sexes, mostly during the breeding season. Pituitary gland obtained from the freshly killed fish from maturing and mature fish or maturity stage 2, 3, and 4 only. Collection accomplished after the cutting, the head by splitting the upper cranium from the lower jaw through mouth using strong and sharp knife. Next, the roof of the cranium cut off horizontally using the pincer and expose the brain. Remove the pea-shaped pituitary gland with fine forceps. Pituitary gland preservation technique. The pituitary gland preserved using acetone drying method. Initially, the gland placed in 10 times their volume of acetone and laterally placed with the new acetone after 12 hours. The acetone changed again after a further 6 up to 8 hours. After these, the gland taken out, dried on a filter paper and stored in tightly sealed bags for further use. Otherwise, we use the freshly extracted pituitary gland immediately. To proceed with the artificial rearing of catfish, we need to have a ripe egg. Thus, we chose a mature female with ripe eggs and anesthetized using the benzocaine or lidocaine. After that, we injected the female the homogenized hormone. The hormones are usually prepared using saline solution or 9% of sodium chloride. It's first ground in mortar and pestle and homogenized with equivalent saline solution in a proportion to the required weight. The female are injected at a dosage of 4 mg pituitary extracted per kg body weight. The injection given intramuscularly between the base of the dorsal fin and the lateral line at the base of pectoral fin or between the anal fin and the lateral line. The hypodermic needle inserted at an angle of 45 degrees and very slowly empties the fluid from the syringe. The injection spot massage gently flowing the withdrawal of the needle to aid distribution of the extract into the muscular and prevent its backflow. The injected female placed into the covered glass aquaria contains fresh and oxygenated water for a length of 14 hours. After 14 hours, remove the injected female and covered the head with a clean towel and dried them, striping the egg into the clean container. During striping, one person hold the tail upward and the other person with wet thumbs slowly squeeze the belly. The next step is extraction of a spermatozoon from the male fish. Before removing the milk, the fish dried with a towel and killed. The fish dissected from the anal opening upward to the chest using sharp knife. The chest is removed with the forceps and put into clean and dried mortar, which are grinded to cover into a milky fluid or milt after cutting into small pieces with a scissor into mortar and crushed by the pestle. A precaution is needed not to mix the milt with water because the spermatozoa only live for 1 minute or 60 seconds after contacting with water. Fertilization becomes reality at the moment the milt are poured into the egg container. A feather or a thin stick is used to properly homogenize them, and water is added to make it more even dense, promoting fertilization. Due to sticky nature of the fertilized egg, we fix them on hairy roots of aquatic grass by dipping the roots into the fertilized egg, placed inside the fresh oxygenated aquarium and covered until the next 24 hours. After 24 hours, we remove the grass root and we see yellow-colored larvae on the glass aquarium floor and dead white eggs on the roots. The larvae have seen processing yolk sac of reserved food, which is green in color. After 72 hours, the larva consumed the most of the stored food and the color changing into dark brownish. During this time, the larva seen with pair of barbels. After 96 hours, the larvae completely consumed the stored food, so extra feed is needed. The extra feed must be live, in most cases it is zooplankton. Tomorrow is another day.